Welcome back to our official YouTube channel from SRN BGNR Government Degree College, Khammam. Myself, Dr. B. Satish Kumar, Assistant Professor in Biochemistry. This video lesson is for final year biochemistry students and most of the life science, of course, all life science courses are having this molecular biology as one of the subjects in their syllabus. And in this video lesson, I am going to explain the translation process. This is the second unit of 6th semester, paper 7. And in this unit, in fact, in the last few classes, we have learned about the genetic code, the features of genetic code, and elucidation of genetic code. We have 3 different experiments which elucidated the genetic code. Okay, so apart from that, you have also learned the structure of tRNA molecule and Obel hypothesis, the degeneracy of genetic code. So this is one of the important topic that we have seen in last or previous class. Obel hypothesis, that is the degeneracy of genetic code. So here we have 61 codons to code for 20 different amino acids. So that is why one amino acid may be coded by one or more different codons. So that degeneracy, there is a process known as Obel base pairing during the interaction of codon and anticodon in translation. Okay, so that explains the degeneracy of genetic code. You have seen that Obel hypothesis in the previous video lesson. Let us quickly go through some of the few previous classes, and let us see the basics of translation. Okay, so when we look at the genetic code, the genetic code is the relationship between nucleotide sequence in mRNA or DNA of gene and the amino acid sequence in a polypeptide chain. So that relationship is called genetic code. So that here 3 nucleotides or 3 bases are used as a code word to specify one particular amino acid. So that is what the genetic code. So we have 64 combinations of codons or code words to specify 20 amino acids, and out of these 64 codons, 3 codons that are UAA, UAG and UGA. So these 3, UAA, UAG and UGA, these 3 are the stop codons that won't specify any amino acids, and that causes termination of translation process. And AUG is the starting codon of all mRNA molecules. So starting codon of the coding region of mRNA. So this is the first codon to be read by the translation machinery, and the methionine is the first amino acid in the newly synthesized polypeptide chain. Okay, so that is about the genetic code and tRNA structure. When we look at the structure of tRNA, we have 4 different arms, and so the main arm is So this one is the anticodon arm or stem and loop structure, anticodon loop. This anticodon loop contains 1, 2 and 3 anticodons. So that means one anticodon that contains 3 bases. These 3 bases in the anticodon specifically recognize the codon in mRNA. Okay, so we will have a mRNA and 1, 2, 3, and this tRNA anticodon interacts with the codon, forming base pairs. Okay, so and at the opposite end we have this amino acid attachment site, and the end 3' end sequence is CCA sequence for all tRNA molecules. At the OH 3' OH of the terminal A, the amino acid will be acylated with its COO group, carboxy group, sorry, carboxy group. Okay, so and two more loops are there. These are for the binding of tRNA to ribosome during translation process. So we have DHE loop, dihydrouridine loop, and we have that tCC loop, this tCC loop, pseudoiodine loop. So and extra arm is also there. Okay, so this is about the structure of tRNA. And Obel hypothesis. In the Obel hypothesis, we have learned about how the degeneracy of genetic code takes place. So the Obel position here, this is the codon and this is the anticodon. So the third base is the Obel. The third base in the codon is Obel position, and the first base in the anticodon that interacts with the third base in the codon. Okay, the interaction between these two is not perfect, so that is why it doesn't obey the Watson-Crick base pairing rules. Whereas in case of the first two bases of codon and last two bases of the anticodon, they base pair and obey the Watson-Crick base pairing rules, strict Watson-Crick base pairing rules. So there is no choice for them. And if you have A in codon, definitely anticodon must have the U. If you have G, then it should have C. Okay, so but in the case of the third base, there is a flexibility. Okay, so that flexibility that was covered in this table. If mRNA, the Obel base is G, then C can recognize it. So if the tRNA is having A, so it recognizes only U. But in case of G, if it is having G, it can recognize either C or U. So that is why it can recognize two codons. It can recognize two more codons, but here inosine can recognize three codons. Okay, so here you can see in this example that leucine here, the tRNA is having U. Okay, the tRNA is having U here. In normal condition, normally it base pairs with A in the Obel position of the codon. But in fact, because of the Obel nature between the interaction between these two bases, either it may interact with A or G. Why G? Because G is also a purine. A is a purine, and instead of A, it can interact with G, because some structural similarity is there between A and G. Both belongs to purine class. So here, and if you have, so that means same tRNA, a single tRNA is recognizing two different codons and specifying the same amino acid. Okay, and in case of glycine, the tRNA is having inosine. Inosine can recognize U, C, and A also, not the G. So that is why it can recognize three different codons and specify only glycine. So therefore, this tRNA is capable of reading three different codons of glycine. So these three different codons are known as synonymous codons, which specify only the same amino acid. So that is why they are synonymous codons. So therefore here we require at least 32 different tRNA molecules to specify 61 codons. Okay, so that is about the translation process. The genetic code, and next topic is the translation process. In the translation process, translation is nothing but protein synthesis. Okay, so from mRNA, the protein will be synthesized. Okay, the protein synthesizing machinery, the translation machinery, is capable of reading that nucleotide sequence in the mRNA and synthesizing amino acid sequence, synthesizing a polypeptide chain with a specific amino acid sequence based on the information present in the mRNA. Okay, so with that means that protein synthesizing machinery can translate the nucleotide sequence into its amino acid sequence. So that is why it is known as translation. So one language is nucleotide sequence language and another language is amino acid sequence language. Okay, so the machinery is translating that nucleotide sequence language into amino acid sequence language. Okay, so for that we require some of the basic components in the during the translation process. Okay, so first one is that mRNA is a messenger RNA which carries message or which has the information for the synthesis of a polypeptide chain. Okay, so it is synthesized from a gene by transcription process, and during the translation process the information in the mRNA is decoded and it is converted into amino acid sequence in protein or polypeptide chain. Okay, so we need mRNA, definitely that information must be there to synthesize a particular polypeptide chain. Okay, and then the tRNA is also very very important, because the tRNA is the adapter molecule which read the codons in mRNA and specifies amino acids in the polypeptide chain. So this is the adapter molecule, this is the real translator that recognizes both the languages. It recognizes nucleotide sequence with one end and it recognizes the specific amino acid with the other end. So that is why it is known as an adapter molecule. So tRNA with one end, that is anticodon arm, it recognizes the codon, specific codon, and then with the opposite end, that is amino acid attachment site, amino acid accepting arm, that carries the specific amino acid that is specific to the codon on the mRNA. Okay, so if anything goes wrong, suppose if the tRNA is specific for phenylalanine and if we add serine on the tRNA instead of phenylalanine, then what happens? The phenylalanine codon now codes for serine instead of phenylalanine. So that means the translation, okay, the translation is the tRNA is key molecule, is the key molecule during the translation process. Okay, so the genetic code is the property of tRNA. So if tRNA doesn't follow the genetic code, then everything goes wrong. So that is why here the genetic code lies within the tRNA. Okay. So and third one is of course the ribosomes. So we have looked at the structure of ribosomes in the previous class. So ribosome in case of prokaryotes we have 70s type of ribosomes, and that ribosome is the protein factory where the remaining molecules like mRNA, tRNA comes and binds to ribosomes, and the ribosome moves along the mRNA and read each and every codon, and during that process tRNAs can specify amino acids based on the codons. So we will get a polypeptide chain. Okay, that is the property of ribosomes. Okay, so these are the prerequisites for the translation process. So in the translation process we have 4 different stages. So we are talking about translation in prokaryotes, not in eukaryotes. So it is prokaryotic translation process where we have 4 different stages. Of course in eukaryotes also the same 4 different stages are there, but different protein factors will be involved. uh, Protein factors will involve at different stages. So there are a lot of similarities between prokaryotic translation and eukaryotic translation, but also some differences are there. Here in our syllabus we have only translation pertaining to prokaryotes only. Okay, so in the prokaryotic translation process we have 4 stages. The first stage is activation of amino acids. Activation of amino acids means the amino acids must be acylated or attached to their corresponding tRNA molecules. Okay, a particular amino acid must be attached to its specific tRNA molecule only. Okay, so suppose one tRNA is there, that is methionine tRNA, that recognizes AUG codon. So then the tRNA must be acylated with methionine only. Then only it reads AUG and specifies methionine at that position. So that is why specifically the amino acid must be attached to the amino acid attachment site of the tRNA at the 3' end. Okay, you have at the 3' end you have CCA sequence, and the end or the terminal base is A. So the nucleotide is A. So that nucleotide is having 3' hydroxy group. At the 3' hydroxy group the amino acid will be attached. Okay, or esterified. Okay, that is what the activation of amino acids. Amino acid must be activated or attached to tRNA molecule. Okay, this is also known as charging. The tRNA is charged by its amino acid. If there is no amino acid attached to a tRNA molecule, so that tRNA molecule is called discharged or uncharged tRNA. Okay, and the second stage is chain termination. Chain initiation. It is chain initiation. So in the chain initiation, what happens? The assembly of all these required components of translation machinery takes place, like ribosome, mRNA and tRNA. Initiating tRNA takes place. Sorry for the interruption. And here during the chain initiation, the initiation complex is formed. During the chain elongation process, what happens? the ribosome gradually read each and every successive codons and that incorporate specific amino acids into a polypeptide chain. So a polypeptide chain will be emerged from the ribosome as the ribosome reads mRNA from 5' to 3' direction. Okay, chain termination. So chain termination also takes place once the entire length of coding sequence has been read by the ribosome. At the after the last codon is read, the then it encounters a termination codon, one of the termination codon. Out of 3 termination codons, one of the termination codon will be there at the end site. So because of the termination codon, the translation process will be terminated because there is no specific tRNA molecule which can recognize the termination codon. So these are the 4 basic stages in the translation process. Okay, so now let us start with the first stage, that is activation of amino acid. Okay, so this is one of the important stage, and this question is also important in case of examinations, because here the specific tRNA, so suppose this is the basic structure of a tRNA, so this is the basic structure of tRNA, and we have anticodon loop at one end and amino acid accepting or at the other end. Okay, so now what happens here, this anticodon, based on the anticodon, a specific amino acid must be attached to this A. A means the ribose sugar of A, 3' hydroxy group of the ribose sugar of A. So now a amino acid will be attached, an amino acid will be attached here. So this attachment requires an enzyme. That enzyme, it is very important enzyme, that is aminoacyl tRNA synthetase. So this is the enzyme which recognize specific tRNA molecule and its specific amino acid. So then it adds that specific amino acid to that tRNA molecule. So that means the genetic code is the property of the aminoacyl tRNA synthetase. So this is the key enzyme which determines the genetic code. If aminoacyl tRNA synthetase makes any mistake, so then that mistake will be reflected in the tRNA, and that tRNA directly incorporates into incorporate the specific that amino acid, mistaken amino acid or other amino acid into the polypeptide chain, that leads to change in the sequence. So that is why here to establish the genetic code, the aminoacyl tRNA synthetase is very important one. So that is why it is also known as second genetic code. Okay. So that genetic code is established by the aminoacyl tRNA synthetase, which interacts with tRNA and its specific amino acid. Okay, specificity is the property of aminoacyl tRNA synthetase. Okay, so for 20 different amino acids, we have 20 different tRNA synthetases. Okay, so that means a specific aminoacyl tRNA synthetase, suppose that is suppose alanine or leucine, for example. Leucine aminoacyl tRNA synthetase is there. So that aminoacyl tRNA synthetase binds leucine amino acid, and it also binds the corresponding tRNA, the leucine carrying tRNA. Okay, so we have three tRNA molecules to carry 3 different tRNA molecules to which can carry leucine. So in that way, that aminoacyl tRNA synthetase can bind any one of these three tRNA. So that is why we require only 20 different amino acid tRNA synthetases. Okay, suppose if you have amino acid tRNA synthetase like this, okay, with one of the subunits, one of the active site, it binds a amino acid. With another uh, portion of active site, it binds the tRNA molecule. Okay, it binds the tRNA molecule. So now what happens? It catalyzes the esterification of esterification of amino acid onto tRNA. So now the tRNA will be acylated, and we will get a charged tRNA. So this is what the function of aminoacyl tRNA synthetase. Okay, so now suppose this is the one specific tRNA, and now what happens? Aminoacyl tRNA synthetase now carry out a specific esterification reaction. Because of that, this amino acid will be attached to the 3' end of the tRNA, 3' hydroxy end of the tRNA. Okay, so this is the amino acid. So now you have the OH and COO minus. Okay, the bond formation takes place between this OH and this O minus. So that is why you have a bond here. So this is an ester bond. Okay, this is also known as acylation. Acid is attached, amino acid is attached, so that is what it is acylated. So now this is called aminoacyl tRNA, a normal tRNA, and this is aminoacyl tRNA. So for this we require that specific aminoacyl tRNA synthesis. Suppose this amino acid is arginine, so then arginyl aminoacyl tRNA synthetase. It is glycine, suppose, so then it is called as glycinyl aminoacyl tRNA synthetase. So that way we will have 20 different aminoacyl tRNA synthetases that can esterify all the tRNA molecules with their corresponding amino acids. Okay, 20, minimum 20 are just enough to specify, to attach or esterify the 20 different amino acids to their corresponding tRNA molecules. Okay, so here a ATP molecule is being utilized. In fact, it is utilizing two molecules. This is equivalent to two ATP molecules. Okay, because ATP is hydrolyzed to AMP plus PPI. Okay, so that is why it requires two ATP molecules for attaching one amino acid onto its corresponding tRNA molecules. So that is why it is known as synthetase. If we write synthase, so that means there is no involvement of ATP. So if ATP is involved, then we write synthetase. Okay, aminoacyl tRNA synthetase. So simply it is aminoacyl, so that means amino acid is being acylated to what? The tRNA. So aminoacyl tRNA synthetase. So this is the synthetase enzyme. Okay, so now during the reaction mechanism of aminoacyl tRNA synthetase, first of all, amino acid will be activated by ATP. So there are two steps in the mechanism of aminoacyl tRNA synthetase. Okay, so in the first step, that specific amino acid, so that means the aminoacyl tRNA synthetase binds amino acid and ATP also. It binds these two molecules. specific amino acid and ATP molecule. So now what happens? It will transfer AMP onto the carboxy end of the amino acid. Okay, carboxy end of the amino acid, and PPI will be released here. So now we have aminoacyl AMP. Okay, at the end of the COO minus, that O minus now is attached to AMP, adenosine monophosphate. Okay, so therefore the amino acid is being activated by the binding of AMP. So that bond between amino acid and AMP now it is a high energy bond. Okay, so therefore amino acid is activated. So now it binds the aminoacyl tRNA binds the specific tRNA molecules, and then this amino acid is directly transferred to the 3' end of the tRNA, specific tRNA molecule. Okay, and releases AMP. Okay, so that means the amino acid never directly acylated onto the 3' hydroxy of tRNA molecules. First of all, it should be activated by AMP, and then it is attached to the 3' end of the tRNA replacing AMP. Okay, this is the mechanism of activation of amino acid. Okay, and so this is activation of amino acid. So in this way, before before beginning of the translation process, we require all the tRNA molecules which are charged with their corresponding amino acids. Okay, so there must be the amino acids charged to their corresponding tRNA molecules. They must be ready for the translation process. Okay, so directly that amino acid never go into the polypeptide chains, and it should be carried by the tRNA, specific tRNA molecule, and that specific tRNA molecule requires aminoacyl tRNA synthetase activity for the esterification of a specific amino acid. Okay, so then only it is possible. So in that means, for the activation of amino acids, we require aminoacyl tRNA synthetases, 20 different aminoacyl tRNA synthetases for 20 different amino acids, and minimum 32 tRNA molecules that recognize 61 codons, and 20 different amino acids, and ATP, of course. So we have finally four requirement in this step. So first one is 20 amino acids, and second one is 20 aminoacyl tRNA synthetase enzymes, and third one is that all tRNA molecules, that is minimum 32 tRNA molecules according to the Obel hypothesis, but there are more than 32 tRNA molecules will be in different organisms. Okay, minimum is 32 number. And fourth one is the ATP, the energy must be provided for the esterification reaction. So these four are essential requirements for the during the activation of amino acids to their specific tRNA molecules. Okay, and in the next class we are going to finish the remaining three stages, that is chain initiation, chain elongation, and chain termination also. And before that, in fact, once again I will revise the structure of ribosomes. In case of prokaryotes, you have this 70s type ribosome, and so we have that small subunit and large subunit. Small subunit contains 16S type of rRNA and some proteins. This 16S is very important. It should be remembered. We have to remember this the type of rRNA which is present in small subunit and large subunit also. So a large subunit will always have two different types of rRNAs in case of prokaryotes. Okay, first one is 23S RNA, second one is 5S rRNA. Okay, so therefore we have three different types of rRNAs to form the ribosome, the 70S ribosome, or the two subunits of 70S ribosome. Okay, this is what the structure of ribosome. Thank you.